So, of course, my father was a very great advocate for science. He loved science, and he communicated that very well. And I was therefore prepared, at least for the possibility of pursuing a career in science. At the same time, he was very careful not to lead me in any particular direction. And I also was very interested in, I very much enjoyed music and literature. When I went to university, I chose for my major subject English literature. And I anticipated a career in that subject. At Harvard College, where I was an undergraduate student, there were many scholars of English literature, scholars who concentrated on particular authors. There was one scholar who focused entirely upon Middle English and the work of Geoffrey Chaucer. There was another who focused, as you might imagine, on the work of Shakespeare. And there were others, again, who specialized in that manner. And so I studied with each of them. And then, as an undergraduate student there, it was also appropriate, it was required, in fact, to do a special tutorial with a one-on-one relationship to a member of the faculty. And for that, it was expected also to write oneself an original analysis of some English, some well-known published work. I did all of that. Nevertheless, perhaps because of the background that I alluded to, I studied other subjects. In the United States, the system of education is different from most of the rest of the world. In America, the emphasis is upon general education, even though it is expected that one will emphasize a subject, such as I did with English literature. It is also required to study many other subjects. And for that reason, I studied history, I studied political science, I studied law, I studied music. In every case, 1 year of coursework in very many different areas. I also, in that spirit, studied chemistry and physics. And I discovered that chemistry had a particular appeal for me. And one day, at nearly the end of my undergraduate study, I woke up and I decided I wanted to continue not in English literature, but in chemistry. I can't explain why. And the rest of my life followed from that moment of decision to pursue chemistry. So Aaron Klug was another important influence on my scientific development. There were several major influences. My father and other very significant figures at Stanford University, where we lived, where I then did graduate study after my undergraduate experience at Harvard. And there my mentor was a great chemical physicist named Hardin McConnell. I pursued my graduate study in the subject, in the area of chemical physics, under his direction. And he was a theoretical chemical physicist. I, however, wished to perform experimental work. And so I didn't take direction from him, and yet he was a powerful influence on my approach and my way of thinking. In particular, it was because of his appreciation of the importance of chemical structure that I decided after graduate study to go to the Laboratory of Molecular Biology in Cambridge, which at the time was really the most active and important place in the world for the investigation of chemical structure, especially as it relates to life science. The first major discoveries in chemical structure related to life science were made in that laboratory only 3 or 4 years before I began my study there. The people who did that work, Max Perutz, John Kendrew, were active. I chose, however, for a postdoctoral advisor, rather than to work with Perutz or Kendrew, to do so with Aaron Klug, because his approach was especially imaginative and far-reaching. It was interesting when I went to work with Klug. I was not attracted to the subjects of his research. They were the structure of transfer RNA and the structure of tobacco mosaic virus. I looked for something else to do, nevertheless, with guidance from him. And a friend called my attention to a publication that had appeared in the journal, the magazine Nature, only a few months before, by Francis Crick, another very well-known, world-leading structural biologist responsible for the discovery of the structure of DNA. I read in that paper about the problem of chromosome structure. I went to Klug and suggested I might study that instead of transfer RNA or tobacco mosaic virus. He agreed. I pursued the problem. It was of great interest around the world, but still an unsolved problem. And it was my good fortune, after a relatively short time, about a year, to discover the solution of that great problem. When I presented that to Klug, he did not accept my proposal for the solution. He told me that it could not be correct. I presented it to Francis Crick. He told me the same thing. He said it was so unlikely and probably incorrect. Again, I was fortunate that it turned out to be exactly right, and it launched my own career. The publication of that work did not make me popular with others around the world who were studying the subject, who believed otherwise. But eventually it proved to be true, and it proved to be fundamental. And the work I do until this day stems from that discovery I made at the age of about 25 years old, very long ago. It was one of the most exciting and important events of my life. And as I say, instrumental in my research activity at this very time. So, on the one hand, it was distressing to be accused of every crime in the scientific book on the one hand, but on the other hand, I was so confident of my solution to the problem that I was never in doubt about the outcome. And I think that it was, on the one hand, important also for the approach to science during the rest of my career. I always wanted to study another large unsolved problem. Mm-hmm. On the other hand, it was also problematic because that is invariably difficult to do. Mm-hmm. And after that work, I suffered a period of some years when I couldn't identify, or if I did identify a problem, couldn't solve another great problem. It was only probably another almost 10 years later before I discovered a new direction. Mm-hmm. And it was for that work, not my original discovery, that I won the Nobel Prize. I think absolutely anyone you would ask would say that my original discovery was the most important and I think the most paradigm shifting in the subject. Mm-hmm. But it was for the later work, not closely related, mm-hmm. that I eventually won the prize. Yes. So the work has, over the course of my entire career, has been directed towards that purpose, to understand how information in the human genome is put to work, mm-hmm. for the development of a creature made of 200 different kinds of cells and tissues, mm-hmm. and the activities of every one of those parts of our organism. It's one thing to record the information as it is done in the genome. Mm-hmm, mm-hmm. It's quite another thing to express it in the full flower of the organism. That is a problem which has also attracted great interest. It has been a central theme in biological science from the very discovery of the genetic code, mm-hmm. to the present day, and it is still a largely unsolved problem. Mm-hmm. My contributions were first to discover the mechanism. How is the information literally read word by word from the genome? Mm-hmm, mm-hmm. And how is the use of the information controlled? We know now precisely the steps of reading, mm-hmm. and even more important, we have, in work up to the present day of my own research group, mm-hmm. a better and better understanding of how it is controlled. The control is key. Mm-hmm. As I told you, there are 200 different types of cell in the human body, mm-hmm. but the same information in every cell. what is responsible for the difference in the types of cell? If nerve cells contain the same information as blood cells, and the same information as muscle cells, then what is responsible for the fundamental difference between them? And the answer is the selective use of the information. Nerve cells only read the part of the genetic book that tells about the formation and function of nerve. Blood cells likewise. Our work has contributed to the discovery of the controlled use of genetic information. In particular, and perhaps most important, we discovered the existence of a molecular computer, an object of great size and complexity, which receives information from everywhere inside the cell and from the environment, including from neighboring cells, and then directs the readout appropriately for the particular cell type, the place, the time in the life of the organism. So it is absolutely true that my thinking and approach and experience of science is shaped by by now very many years of the practice of science, but possibly in a different way than what you may think. So I haven't changed in my orientation.